Hello everyone, I am K. Devavani, Assistant Professor of Microbiology from Government Degree College for Women, Nalgonda. Today I am going to present a topic on phagocytosis. The contents here I am going to present are the definition of phagocytosis and the cells of phagocytosis and the mechanism of phagocytosis, the enhanced phagocytosis process, that is opsonization, and the functions of the cells of phagocytosis. Phagocytosis. It is derived from Greek phagein, that means to eat. Cytos means cell. Osis means it is a process. So the meaning clearly explains that the process used by the cells to eat the foreign particles or dead cells or any harmful particles which are injurious to the host. The process is called phagocytosis. Here it is a process of eating here in the form of engulfment and digesting in the form of destruction of those harmful particles, which particularly are bacteria, dead tissue, and any other foreign particles by the cells. The process of killing by engulfing is simply termed as a phagocytosis process. So the phagocytosis is carried out by the cells. Those cells are involved in this phagocytosis process are called as phagocytes. The cells involved in the phagocytosis process are called as phagocytes. The phagocytes are first discovered by Elie Metchnikoff in 1883, and he described these phagocytes as two types: microphages and macrophages. Microphages and macrophages. Microphages here are neutrophils. Neutrophils are considered to be microphages, and macrophages here macrophages. Let us see the structures of these cells which are involved in the phagocytosis process. So first one is neutrophils. Neutrophils are also called as PMLs, polymorphonuclear leukocytes. PML means polymorphonuclear leukocytes. These neutrophils are major and they are abundant in the blood and nearly constitute about 50 to 70% of the circulating leukocytes. Neutrophils are multilobed and contains 3 to 5 lobes joined by strands of genetic material, that is chromatin. And the cytoplasm contains granules. Primary granules are also called aerophilic granules, and the cytoplasm also contains secondary granules which are rich with certain enzymes like gelatinase, lysozyme, and collagenase. They originate, all these phagocytes, they originate from the myeloid progenitor cells during hematopoiesis. There are two types of progenitor cells: lymphoid progenitor cells and myeloid progenitor cells. Myeloid progenitor cells, they give rise to red blood cells, white blood cells, platelets, etc. Whereas lymphoid progenitor cells, they give rise to lymphocytes. They are T, B, and natural killer cells. These phagocytes, these neutrophils here, they originate from this myeloid progenitor and they differentiate in the bone marrow and they are released into the blood before migrating into the tissue, where they have a lifespan of very few days. These neutrophils, they do not have rough endoplasmic reticulum, and the nucleolus, it disappears with the as the cell matures, and these cells are recruited to the site of infection. Whenever there is an infection by the pathogens, these cells are moved to the site of infection. How they move means they respond to the certain chemical signals that are released by pathogen at the site of infection. This process of movement towards to the site of infection is also called as a chemotaxis, and these are the dominant first cells that respond to infection, and they are the main components of the pus. Briefing about neutrophils. Neutrophils are multilobed, contain 3 to 5 lobes, which are joined by genetic material, and it contains primary and secondary granules rich with enzymes, and they originate from the myeloid progenitor cells and they move into the tissues, and these are the first respondents to the infection, and they move towards the site of infection. And here these cells do not contain rough endoplasmic reticulum, and the nucleolus disappears with the as the cell matures. And another important one is monocytes. Monocytes are the largest leukocytes, constitute about 5 to 10% of the white blood cells, and these monocytes, they appear in amoeboid shape, and they do not contain any granules, so they come under the category of agranulocytes. WBC here are granulocytes and agranulocytes, so monocytes, they come under the agranulocytes, and it contains a single lobed nuclei. This may be either ellipsoidal or it may be in a bean shape. Monocytes, generally they are categorized into two types: inflammatory monocytes and patrolling monocytes. Inflammatory monocytes are those, they enter the tissues quickly, very quick in response to the infection. But whereas patrolling monocytes, they are generally they crawl along the blood vessels, they are present along the blood vessels, and they provide as a reservoir for the tissue monocytes in the absence of infection. These monocytes, when they migrate into the tissues, and they differentiate into macrophages. Monocytes are changed into macrophages in the tissues. During this change, the changes occur, or the cell increases, and the organelles also increase in number that are present in them. Eosinophilic granules are present, they also increase in number and also produce high levels of hydrolytic enzymes. Once they reach into the tissues, they are called as the macrophages. Macrophages are the histiocytes. Histiocytes means the monocytes in the connective tissues. So macrophages are histiocytes, which are the wandering amoeboid cells seen in the tissues. These monocytes and macrophages, they are also originated from the myeloid progenitor cells. The major function of macrophages here is antigen presenting cells, are also antigen processing cell. So here APC, antigen presenting cells or antigen processing cells. What is the meaning for antigen presentation or antigen processing? These cells, they capture the antigen and they process it and they make it into smaller fragments during the phagocytosis process, and these fragments are now called as antigenic peptides, and these antigenic peptides are presented onto the surface of the membrane along with the MHC that is present on its surface. Major histocompatibility MHC2. Why? Because when the antigenic peptides are presented with MHC2, then only they are recognized by the T cells. For the activation of T cells, the antigenic peptides should be presented with the MHC2. So that processing is called as the antigen processing or antigen presentation. So the major role of macrophages here is phagocytosis and the antigenic processing. The resting macrophages need to be activated for the phagocytosis. After reaching into the different tissues, they are named differently, like the macrophages that are present in the connective tissue are called as histiocytes, and the macrophages that are present in the bone are called as osteoclasts, and the macrophages that are present in the central nervous system or brain is called as microglial cells. The macrophages in the lungs are termed as alveolar macrophages. The macrophages in the liver are Kupffer cells, and in the kidney they are called as mesangial cells. So they have different names based on the different presence in the different tissues or organs. The major function of macrophages here is phagocytosis, able to engulf the foreign particles and internalize them, and the second one is antigen presentation, and the third one is production of cytokines, particularly IL-1. So macrophages, they serve the bridge between the innate and adaptive immune response. Innate immune response is non-specific immune response, generally acquired from the birth, but adaptive immune response is a specific immune response. So for these two immune responses, the macrophages serves as a bridge. It is the connecting link between these two immune responses. Macrophages are the first line defense mechanisms for the infections and the caused by the microorganisms. Now the last one is the dendritic cells. Are first described by Ralph Steinman in 1970, and he received the Nobel Prize for it. And these cells, they contain the long membranous extensions and they resemble to the structure of neurons, so they are named as dendritic cells. Generally, these dendritic cells are present in those tissues which are in contact with the external environment, like skin. In the skin, these cells are called as Langerhans cells, and other linings of the nose, lung, stomach and intestine. Major function of these dendritic cells is antigen capturing, antigen processing. These cells, they capture the antigen at one location and they present this antigen at another location is by means of three ways. That is, one is phagocytosis and the second one is pinocytosis. Pinocytosis means engulfing a fluid liquid, and receptor mediated endocytosis. By means of these three ways, the dendritic cells process the antigen and present this to the T cells. The immature dendritic cells are also called as wheeled cells. Wheeled because it contains the cytoplasmic wheels. So they are immature. Immature dendritic cells are also called as wheeled cells. So these are all the cells. These are the cells involved in the phagocytosis process. So let us move on to the mechanism of phagocytosis. How these cells they engulf and they kill the foreign particles or the bacteria or any harmful particles. So it includes steps. They are, first one is here the chemotaxis, and the second one is ingestion, third one is the formation of phagosome, and fourth one is the phagolysosome formation and the digestion, and it is followed by the exocytosis. So here let me explain this mechanism through different steps. The first step is chemotaxis or activation. The resting phagocytes need to be activated. What are the activating signals here? Means they are the inflammatory molecules. They are the molecules of microorganisms, cell surface structures that trigger that activate these resting phagocytes. Those cell surface structures generally are the proteins or the capsules or the murine or so on. So these inflammatory molecules they activate the resting phagocytes which are in rest, and now they gain the ability to enter into the tissues towards the site of infection. Based on those signaling molecules, inflammatory molecules, macrophages are these neutrophils, they move to the site of infection. That process of movement of these phagocytes towards these chemical molecules, that movement is called as a chemotaxis. Chemotaxis is the movement of phagocytes towards the chemotaxins. What are chemotaxins? Here they are the inflammatory molecules produced by the bacterial products or the injured tissue. The second step here, now they reach to the site of infection. Now these leukocytes they leave the capillaries and they need to enter into the tissues. So for that purpose they take up the marginal positions of the blood vessels and they stick on to the venules of them and they roll along with the vessel wall, and this is called as an attachment. Now they begin to move out of the blood vessel here. They produce an enzyme collagenase to digest the membrane where they attached, and by means of this enzyme they from that place they come out of the venules. Now the phagocytes they should recognize the pathogens. So for this recognition the phagocytes recognize specific conserved motifs which are present on the microbes, bacteria, fungi, parasites. They recognize the conserved motifs. Those conserved motifs are called as PAMPs. PAMPs means pathogen associated molecular patterns. Pathogen associated molecular patterns. So after coming out of the vessels, they to recognize and to bind to the microbes, they recognize a conserved motif that is present on the surface of the microbe, that is called as a pathogen associated molecular patterns. These PAMPs now they bind to the phagocytes. So the receptors that are present on the phagocytes, now the receptors that are present on the phagocytes are called as PRRs. it is a microbe or any harmful particle, foreign particle. This it should bind to the macrophage. The receptors that are present in the macrophages are the phagocytes are called as PRRS, whereas on the microbe it has PAMPS. So on microbes they contain PAMPS, to which the PRRS bind. PRR means pattern recognition receptors. PRRS means pattern recognition receptors which are present on the phagocytes. This is one mechanism of binding the phagocytes to the microbes. Phagocytes are also get activated by the antibodies and the complement proteins, because the phagocytes they have the receptors for the binding to the immunoglobulins or the antibodies and to the complement proteins. This process is called as opsonization. If the antibodies or complement is involved to bind to the macrophages or the phagocytes, then that process is called as opsonization. So opsonization is again, it is derived from Greek word. Opsonization means it is tasty. Like this, now the microorganisms and the phagocytes are recognized by their receptors and they are bound to each other. Now the third step, that is a endocytosis process. This binding of microbe to the phagocytes by this PRR, pattern recognition receptor, or the opsonins, they activate the pathway, they activate the signaling pathway that trigger the actin polymerization. This polymerization in turn results the extensions from the membrane in the form of pseudopodia like around the microbe, and it forms like a structure that phagocytic vesicle. It forms it's a vesicle structure, vesicular structure. Binding of microbe to phagocytes by PRRs, pattern recognition receptors, or the opsonins, they activate the signaling pathway that trigger the actin polymerization. This actin polymerization that leads to the membrane extensions and form a vesicle around the microorganism. This and that is taken inside, internalized. Now that structure of vesicle, phagocytic vesicle, is called as a phagosome. Now the phagosome, it fuses with the lysosomes that are present in these phagocytic cells, and the granules in the neutrophils, they are the granules, they fuse with them and form a structure that is called as a phagolysosome. Now the phagosomes, they fuse with the lysosomes. It forms a structure that is called phagolysosome. Here in this phagolysosome, antigens or these foreign particles will be digested or they are fragmented by following any one of the, because they contain antimicrobial agents. Those antimicrobial agents may be proteins or peptides, defensins and catalycidins, or contain the agents low pH. They contain low pH and acid activated hydrolytic enzymes such as lysozyme and proteases, and also contain some specialized molecules that mediate the oxidative attack. So phagolysosome contains antimicrobial agents that may be of antimicrobial proteins and that can activate the enzymes with the low acid conditions, and specialized molecules that can mediate the oxidative attack. So here in this phagolysosome, the pathogens or the foreign particles are killed. That killing is follows either by means of two ways. One is lipases, nucleases, lysozymes, which are present in these lysosomes, or they make it into fragment. They kill it. Another way is oxidative attack, which is highly toxic Because in this oxidative attack, reactive oxygen species means here ROS. Another one is RNS. Reactive nitrogen species will be generated. So in the phagolysosomes, the microbes are killed either by the oxygen dependent mechanism or by the oxygen independent mechanism. The oxygen independent mechanism involves the enzymes that are present in the lysosomes, and oxygen dependent mechanism is also called as oxidative burst, where it leads to the generation of reactive oxygen species and reactive nitrogen species, and they are unique to the phagocytes by an enzyme that is called as a NADPH oxidase. The enzyme is a NADPH oxidase. So reactive oxygen species generated by the phagocytes activates an enzyme that is NADPH oxidase. This enzyme, once it is activated, it converts the oxygen into superoxide radicals. Here again, the superoxide radicals are converted into hydrogen peroxide by the enzyme superoxide dismutase, and also this enzyme produces another reactive oxygen species, finally, which are highly toxic to the microorganism, and finally kill it. Another one is reactive nitrogen species. They are generated by the transcriptional activation of a gene, means it is an inducible gene, so that is activated for the enzyme. That enzyme is a nitric oxide synthase, that is also called as a INOS. I means inducible nitric oxide synthase. So this enzyme now produces NO, that is the nitric oxide, which is a potent antimicrobial agent. So by means of these two processes and two ways, the microorganisms or the antigens are killed and they are fragmented here in the phagolysosome. So now this is partially digested, and digested microbe, now that leads to a residual body and indigestible material. The residual body, here it contains indigestible material. That material is released outside by the process of exocytosis. So this is the whole mechanism of phagocytosis by the phagocytes. So the different steps here is, first one is chemotaxis, movement of phagocytes towards the site of infection, and here after reaching to the site of infection, they recognize the receptors, PRRS, pattern recognition receptors. So once they are bound, that leads to the activation of signaling pathway, leads to the polymerization of actin. It leads to the extensions of membranes in the form of a vesicle, leads to the formation of phagosome. Now this phagosome fuses with the lysosome and forms a structure, phagolysosome. Here the killing occurs by means of oxygen dependent and oxygen independent mechanism is by means of the enzymes that are present in them, proteases, phospholipases, nucleases, lysozymes. They are involved in the killing the antigen, and oxidative burst, taking the oxygen, here produce reactive oxygen species, reactive nitrogen species, which are highly potent antimicrobial agents. So by and the undigested material, that is called as the residual body, that is released outside, that is in the form of exocytosis. So this is the basic mechanism of phagocytosis shown by the cells of phagocytes. Next is the opsonization. As mentioned earlier, the phagocytes are also activated by the antibodies. Here the antibodies are immunoglobulin G, that means immunoglobulin G, they activate. And the complement proteins here is a C3b and C4b, the complement proteins. Antibodies, they bind to the antigens, and this in turn binds to the macrophage, and antigen antibody complex activates the complement proteins, and these complement proteins again they bind to the phagocytes. So immunoglobulins and complement proteins, they enhance this the process of phagocytosis. So the killing is more efficient than the phagocytosis. So this is called as enhancement of phagocytosis, is called as a opsonization. So here in this picture you can see that this is an opsonized, it is an antigen which is bound to this is an immunoglobulin, and these are the C3b, C3, C4b, and which can bind to the antigen. This is called as an opsonized antigen. This bind to the phagocytes. So that leads to the enhancement of phagocytosis. This is called as the opsonization. The major functions of neutrophils here, and the macrophages and the dendritic cells, here is antigen killing by means of phagocytosis, and the dendritic cells and macrophages, they are the major antigen presenting cells. That means they activate the T cells. In this process of killing, the antigenic fragments that leads to the antigenic peptides, those are presented with the MHC2 that are present on the surface for the activation of T cells to activate the cell mediated immune response, that is adaptive immune response. This is about the phagocytosis in brief, and the phagocytes which are involved in the phagocytosis process. Thank you.